Okay, for the second lecture video for Unit 1 in Blood Bank, we're going to talk about the nature of antigens and antibodies and the major histocompatibility complex. And then in a separate video, which will also be due this week, we're going to talk about antigen and antibody reactions and how to grade agglutination reactions. Okay, so this first video, we're going to talk about immunogens versus antigens. And even before I get into that, hold on, I'm sorry, give me 1 second. I'm in the wrong part of my notes. Okay, so immunogens versus antigens. So these are the two key terms used to define substances that are targeted by the adaptive immune responses. So from the first video, we learned about innate and adaptive immune responses and how those differ. So immunogens and antigens are part of the adaptive immune response. So an antigen is a substance that is specifically recognized by the adaptive immune system. So it's this foreign molecule that binds specifically to an antibody or a T cell receptor. And again, just think about, I always think about antigens as being kind of like little, it's a cell surface marker. So I just kind of think about it as being like a little name tag or like a flag on the cell that just kind of identifies it. And the antibodies that our body has built in response to that antigen. So then that's the part that's part of the adaptive immune response. Those antibodies are recognizing that antigen from that molecule. And then an immunogen is a substance that's capable of causing an adaptive response. So all immunogens are antigens, but not all antigens are immunogens. So some of the different factors that can influence the immune response, and we've talked about several of these before in micro. Age, so of course elderly and neonates are more likely to have a decreased immune response, and that's either just from the immune system has not been developed yet or it's starting to degrade. The patient's overall health status, so folks who are malnourished, fatigued, under constant stress are less likely to be able to mount an adequate immune response. Genetic composition, so there's a lot of different genes that influence innate and adaptive immunity, but MHC or major histocompatibility genes have the most impact. So they code for cell surface molecules for antigen recognition. So it's this part of our bodies that help us recognize self from non-self. The antigen dosage, so enough of the antigen has to be presented to meet the lower threshold required for activation of the adaptive immune response. So there has to be enough of that foreign cell or pathogen there in order for our body to be able to recognize it as a threat and then respond accordingly. And then the route of exposure, so the way that we come in contact with the antigen, so is it intravenously, intradermally, subcutaneously, intramuscular, oral. So the way that we come in contact with it could determine how or if our bodies produce an immune response. Okay, so some different traits of antigens and immunogens. In general, immunogenicity is the ability of an antigen to stimulate a host response. So this, the immunogenicity of the antigen depends on a couple of different things. So it's macromolecular size. So you're usually going to have a molecular, these particles are going to have a molecular weight of at least 10,000 Daltons to be recognized by the immune system. The most effective immunogens are greater than 100,000 Daltons, but there are some substances with a molecular weight of less than 1,000 Daltons that have been known to provoke a response. So not only do you have to have enough of them, but they have to be of a certain size. The foreignness of the particle. So lymphocytes are selected during that maturation process that we talked about in the first video, so that they respond only to antigens that are recognized as non-self, or those that are not related to the host, because we don't want our immune cells attacking our own cells. The chemical composition and molecular complexity. So proteins are going to be the most immunogenic type of particle as compared to other substances like polysaccharides, nucleic acids, and lipids. And then the antigen's ability to be processed and presented within those MHC molecules. So is there enough of it? Are the particles big enough? Are they recognized as foreign? What's their chemical composition? So proteins are especially immunogenic. And then is that particle able to be processed by our immune cells, and then presented as a foreign cell? Okay, epitopes. So this is the small portion of the total molecule that's actually recognized by the lymphocyte. So it's a single antigenic determinant. The key portion of the immunogen is known as the antigenic determinant, or epitope. The exact molecular shapes or configurations are recognized by our B cells, or the peptide sequences could be detected by T cells. Larger molecules could contain numerous epitopes, each one capable of triggering the production of a specific antibody, or of activating a T cell. So B cells recognize two different types of epitopes. The first one's called a linear epitope. So you've got sequential amino acids that are on a single polypeptide chain. And then you've got what are referred to as conformational epitopes that result from the folding of one or more polypeptide chains. So it brings together amino acids that would normally be distant from each other, so that they're recognized together, kind of like a pair. So here's a picture of an epitope. So this is your red cell. These are different antigens, or the epitopes. So, and you can think of them kind of like a lock and key type mechanism. So you've got the antigens, or the epitopes that are on the cell surface, and then you've got these little Y-shaped things here recognize or symbolize your antibodies, circulating antibodies, and then the other side of the lock and key mechanism is the part that is specific to the particular antigen, and you can see this is the same shape as what's on the end of the antibody. So this antibody is specific to this antigen. Okay, haptens. So a lot of substances are too small to be recognized by B cells, but they can still stimulate a response if they're combined with a larger molecule. So this is a substance that is non-immunogenic by itself, so it's not able to produce that immune response by itself, but it can form new antigenic determinants if it's combined with a larger carrier molecule. So at that point, it would be able to react with antibody, but not able to stimulate antibody production. Haptens are antigens, but they are not considered immunogens. So when they're bound to a carrier molecule, the hapten is able to initiate antibody production that can react with the hapten, even in the absence of that carrier. So it has to kind of combine with the carrier in order to initiate that response. But then after that point, the antibody can then react with just the hapten. The small size of the hapten and single determinant site makes them incapable of antibody-mediated precipitation or agglutination because cross-linking with more than one antibody molecule is not possible. So we're going to talk more a little bit later about precipitation and agglutination reactions. Those are just when you have multiple like antigen-antibody reactions that are occurring together, and the result of that is visible, a visible product that we can see. So that's the precipitation or the agglutination. And it's happening by those multiple antigen-antibody complexes cross-linking together. Haptens are also found in nature. So catechol are found in poison ivy plants. So once they come in contact with your skin, the catechols join with your tissue proteins to form immunogens that cause that wonderful contact dermatitis. Okay, adjuvants are substances delivered simultaneously with antigens for the purpose of potentiating or enhancing that immune response. So they're able to increase that immunogenicity of an antigen by stimulating your innate immune receptors that detect the presence of an infection or foreign particle, some type of danger that it recognizes to your immune system. They're also thought to work by preventing the antigen from diffusing away from the site of inoculation. So that allows your innate immune cells to accumulate at the site. So it kind of tricks the immune system into acting as if an infection is underway, even if antigen inoculum given to the patient is essentially harmless. A lot of vaccines use adjuvants to achieve that protective immunity against the target antigen. So those that don't are usually made of whole pathogens or substances that come from either killed or weakened pathogens. Okay, the relationship of antigens to the host. So antigens can be placed in a couple of broad categories depending on their relationship to the host. So autoantigens are those antigens that belong to the host. So they're not going to evoke an immune response under normal circumstances, but if an immune response does occur to autoantigens, it may result in autoimmune disease. So again, our body is supposed to not attack its own cells. So that's why we have those. Our immune system has the capability of detecting self from non-self, but sometimes it gets confused. Alloantigens are from other members of the host's species, so they are capable of eliciting an immune response. And then this alloantigens have to be taken into consideration in tissue transplantation and in blood transfusions, because we don't want to introduce an alloantigen or a foreign antigen to the patient because they could have that immune response. Heteroantigens are antigens from other species, so other animals, plants, or microorganisms. And then heterophile antigens are heteroantigens that exist in unrelated plants or animals, but they're either identical or closely related in structure so that the antibodies can cross-react with one another. We're going to talk about the major histocompatibility complex, or the MHC. So HLA, or human leukocyte antigens, is the genetic capability to mount an immune response. So it's linked to this group of molecules that originally were originally referred to as human leukocyte antigens. So they're now known as the MHC molecules because they determine whether transplanted tissue is histocompatible, and if it's going to be accepted by the recipient, or if it will be seen as foreign and be rejected. So these MHC molecules are found on all nucleated cells in the body. They're found on chromosome 6. They play a pivotal role in the development of humoral and cell-mediated immunity that we talked about in the first video. Their main immune function is to serve as carriers of peptide antigens for recognition by T cells. So T cells require antigens to be kind of cradled within the MHC molecules for that recognition step to occur. There are a few different classes of MHCs. So your class 1 MHC is involved in antigen recognition by T cells. It's expressed on all nucleated cells in the body, but there could be some variations based on cell type. Your class 2 MHC is involved in antigen recognition by T cells, found primarily on antigen presenting cells like your B cells, macrophages, dendritic cells, and some T cells. And then your class 3 MHC is kind of completely different from the other two in that it codes for complement and cytokines. So it's not involved in antigen presentation or binding. It's not expressed on the cell surface, so that one's a little bit different. And I would also recommend, there is a YouTube video in Moodle about the MHC complex. I would recommend pausing the lecture here and watching that video. Okay, production of cytokines. So we talked about cytokines in the first video. They are the chemical messengers throughout our body. So they help regulate immunity, and they will influence the innate and the adaptive responses to infection. So they're produced by immune and non-immune cells. There's small proteins that bind to and activate receptors located on the target cells. A lot of them are produced in response to some type of infectious stimuli. Some of the effects of cytokines can include regulation of growth, differentiation, gene expression by a lot of different cell types. So after those cytokines or those chemical messengers are released, most are going to be rapidly degraded in the extracellular environment, so it limits their activity. They're classified according to the distance traveled between the cell that produced the cytokine and the target cell. So they're classified as autocrine, which are cytokines that bind to receptors on the same cell from which they were secreted; paracrine, which are those that act on cells within the tissue region surrounding the source; or endocrine, which are cytokines that diffuse into the bloodstream, and so they're able to influence cells that are far away from the cell or cells that produce them. So in this context, cytokines are acting kind of like hormones. Some cytokines can act alone, some act in conjunction with other cytokines, and we've talked previously in micro about synergistic and antagonistic relationships between organisms and as that relates to drug interactions. So talking about cytokines, synergistic relationships are where the cytokines interact with one another and they kind of complement or enhance the activity of the other. Antagonistic is when they counteract the actions. A cytokine cascade is where you have cytokines that induce the production of additional cytokines by the target cells. So cytokine interactions are very powerful determinants of immunity. So the pattern of cytokines produced in response to infection can be the difference between either an effective or an inappropriate immune response. And then there's something called a cytokine storm, where you have a massive overproduction and dysregulation of the cytokines. So this results in hyperstimulation of the immune response, also referred to as hypercytokinemia, which can lead to shock, multi-organ failure, and can lead to death. So when we were talking in the first lecture video, we were talking about regulatory cells, and so this is why we have that regulation system built into our immune response, because we want it to respond appropriately to those foreign particles, but we also want it to be able to stop that reaction when it's no longer necessary. Okay, this next little bit is a good chunk of information on Antibodies. So just some general information. They're also referred to as immunoglobulins, which are globular proteins made up of about 86 to 98% polypeptide and anywhere between 2 to 14% carbohydrate. They make up about 20% of plasma proteins in healthy people. They can be detected by serum protein electrophoresis, where you've got serum from a patient that's placed on a gel. You run an electrical current through it, and it separates the protein. So if that process is done at a pH of 8.6, most serum proteins can be separated by size and by charge, and you've got 5 distinct bands that you can obtain in that process. So immunoglobulins are the slowest moving proteins. They're going to appear primarily in the gamma band, so they're also referred to as gamma globulins. And then detection of specific antibodies is especially helpful for blood banking, but it can also help to diagnose different diseases. Okay, so the antibody structure, each different class of antibody or immunoglobulin has different distinct properties. All immunoglobulin molecules share a lot of common features, and their basic structure is going to be similar. So each immunoglobulin has 2 heavy chains, which are large peptide chains. The 5 major classes of the heavy chains is what determines the isotype of the antibody. So the heavy chains are bound by 2 smaller peptide chains, which are referred to as the light chains. So there's 2 major types of light chains that exist. You've got kappa and lambda. Antibodies have either 2 kappa or 2 lambda chains. They're never going to have one of each. So the heavy and light chains are held together by disulfide bonds, and the exact number of those bonds differs between the different antibody classes and subclasses. Each heavy and light chain has a single variable region and 1 or more constant regions. So the variable region is going to be unique to each different antibody type, and it's going to allow the molecule to bind specifically to that particular antigen. The constant regions are the same in each of the immunoglobulin classes or subclasses, and they're responsible for different biological functions that play a role in immune defense against an antigen. So a variance in the constant regions of the heavy and light chains are called allotypes, and then idiotypes determine the antigen binding specificity. So what is the specific antigen that antibody will bind to are found only in the variable regions of the heavy and light chains. So they're specific to each antibody molecule. So you can think about it like each antibody is going to have that variability or that variable region where there will be differences that are specific to the antigen that it pairs with. Scoot down here, the FAB antigen binding site. So these are enzymes, and papain can be used to break apart immunoglobulin molecules to produce 2 different fragments known as the FAB, which is fragment antigen binding, and the FC, which stands for fragment crystallizable. The FAB portion contains the part of the molecule that binds to the antigenic determinant, and then the FC, complement binding site. So the remainder of the constant domains of the 2 heavy chains are linked by disulfide bonds. So certain immune cells, like your macrophages and neutrophils, are going to have receptors for the FC portion of the immunoglobulin. The immune cells can bind the FC portion of the antibodies attached to red cells or to pathogens, and they can help with their removal through the process of phagocytosis. The hinge region of the antibody molecule allows for flexibility of the molecule, so it allows the 2 antigen binding sites to operate independently from one another. So not all antibody types have this hinge region specifically, IgM and IgE do not. And the term valency refers to the number of antigen binding sites for any given antibody, or the number of antibody binding sites for any given antigen. So here's, I got a couple pictures included here of the antibody structure. So you've got your variable regions, and all antibodies kind of have this Y shape. Variable regions on the end, so that's the part that's you're going to see variations between the immunoglobulins, and that's the part that's going to bind to the antigen. So that portion is going to be specific to the antigen. But the heavy chain and the light chain, so heavy chain on the inside, light chain on the outside. This is the constant region. And then here, whoops. Here you can see where they're talking about the FAB and the FC, the hinge here. Okay, and then there's a video in Moodle, YouTube video on antibody structure. Might be a good idea to take a look at that one at this point. So I mentioned that there are 5 different antibody classes. So 5 major classes of immunoglobulins based on properties associated with the constant region of their heavy chain. So all are going to be assigned what's referred to as a sedimentation coefficient, sometimes referred to as Svedberg units. So it's a measure of the rate by which the molecule sediments in a high-speed ultracentrifuge. So when you see the S number, so that's the S number that you see associated with each of the immunoglobulins, that's what that's referring to. So the first antibody class is IgG. This is the predominant immunoglobulin in humans. So it makes up about 70 to 75% of our circulating serum or plasma. It has the longest half-life of about 23 days. It is a monomer, so one tetrapeptide unit with a molecular weight of 150,000 and a sedimentation coefficient, or a sed rate, of 7S. It's referred to as bivalent because it has 2 antigen binding sites. And then within each of these immunoglobulin classes, there are some subclasses as well. So IgG has 4 subclasses, IgG 1, 2, 3, and 4. All of these subclasses can cross the placental barrier except for IgG 2. And IgG is the only one of the 5 that can do this. So this is, IgG plays a very important role in providing immunity to the newborn. IgG3 is the subclass that is most able to bind complement because it has the largest hinge region. Then IgG1, then IgG2, and IgG4 have shorter hinge segments, so they're not able to bind or activate complement as well. IgG is an important mediator of opsonization. And if you remember that from the first video, that's the coating of a foreign antigen that leads to enhanced phagocytosis. It's also able to neutralize bacterial and viral toxins, but it's also considered an incomplete antibody, which means that it's not able to agglutinate with an antigen. It only sensitizes the cell. So here's what it looks like, a couple of different variations there in the structure or the size of the different subclasses. Okay, IgM is a macroglobulin, so it's the largest of all of the immunoglobulin classes. It has a sedimentation rate of 19S, molecular weight of approximately 900,000, valency of 10. It's considered a polymer or a pentamer, so it's made of 5 monomeric units that are held together by a glycoprotein known as the J chain. or joining chain. So they form like a star shape. IgM has a half-life of about 5 to 6 days, and it makes up about 5 to 10% of all serum immunoglobulins. It has a lot of the same functions as IgG, but it's more effective because it has those multiple binding sites. It's more efficient at triggering the classical pathway of complement activation and most efficient at agglutination reactions, and that's just because it's larger. And because of its large size, it's mainly found intravascularly, so not in other body fluids or tissues. It also can neutralize bacterial and viral toxins, but it cannot cross the placenta. And it's known as the primary response antibody. So we kind of mentioned this one in the first video. Also, it's the first one to appear after antigenic stimulation. It's the first one to appear in a maturing infant. And a lot of times, because it's the first one to appear, we can measure the rates or the presence of immunoglobulins in a patient's blood, so we can use it to diagnose acute infections, because if IgM is the first one to appear after exposure and the patient has an increased number of IgM antibodies, then we know that they're in the acute phase of an infection. So here's what the IgM antibody looks like. So you've still got that Y shape, but then they're all joined together by that J chain to form this larger structure. Okay, IgA makes up about 10 to 15% of all immunoglobulins in our circulating serum. It's a monomer with a molecular weight of about 160,000, sedimentation coefficient of 7s. It has two subclasses, IgA1 and IgA2. Your IgA1 is going to be found in higher numbers in serum. IgA2 is found in secretions from mucosal surfaces. So it's going to appear as a dimer in respiratory, urogenital, and intestinal areas. The major role of your serum IgA is that it acts as an anti-inflammatory agent. IgA2 found as a dimer in your respiratory, urogenital, intestinal mucosa. It can also be found in breast milk, colostrum, saliva, tears, and sweat. So that dimer consists of, and di, the di indicates two, so it consists of two monomers held together by a J chain. So still similar to that J chain that we see in the IgM molecule that's holding all of those single monomers together. The major role of secretory IgA is it kind of patrols your mucosal surfaces and acts as a first line of defense by preventing antigens from penetrating farther into the body. So we talked about that as one of your natural host defenses in microbiology. So you've got those IgA antibodies in your nasal passages, mucosal surfaces. So it's just the first line of defense against foreign pathogens. IgA is not able to bind to complement by the classical pathway, but aggregation of immune complexes could trigger the alternate complement pathway. But that lack of complement activation could assist in clearing the antigen without triggering that inflammatory process. So because of that, it can minimize tissue damage. Here's what your IgA looks like. So again, you've got that Y-shaped structure. So there's your monomer joined to another monomer by that J chain. So then it's referred to as a dimer. Okay, IgD, present in very low levels in serum, less than 0.001%. It only has a half-life of about 1 to 3 days. Molecular weight of 180. It has a really long hinge region. Sedimentation coefficient of 7s. It really doesn't appear to serve any type of protective role. It doesn't bind complement, neutrophils, or macrophages. It does not cross the placenta. It might play a role in B cell activation, but they don't really know. Most of the IgD found on the surface of most of the IgD that is found is on the surface of immunocompetent but unstimulated B lymphocytes. And there's what it looks like with that very long hinge region there. Okay, IgE is the least abundant immunoglobulin, so even less than IgD in serum, so it only makes up about 0.0005%. Molecular weight 190. It's an 8S molecule. It does not participate in complement fixation, agglutination, or opsonization. It doesn't cross the placenta. It can attach to basophils or Langerhans cells, eosinophils, and tissue mast cells through, it has high affinity receptors that are found exclusively on the surface of these cells. So those antigen binding sites are still free to interact with the specific antigen. Your plasma cells that produce IgE are located mostly in the lungs and in the skin. When you've got two adjacent IgE molecules on a mast cell to bind a specific antigen, a cascade of cellular events is initiated that results in degranulation of the mast cell, which results in release of vasoactive amines like histamine and heparin, which induces what's known as a type 1 immediate hypersensitivity or allergic reaction. So some of those typical types of reactions are hay fever, asthma, vomiting, and diarrhea. Hives could be even as bad as anaphylactic shock, which anaphylaxis is the most rapid form and most severe form of hypersensitivity. And we get much more into hypersensitivity reactions in the last unit. IgE also serves a protective role because it can trigger an acute inflammatory reaction that sends neutrophils and eosinophils to the affected area to help destroy those foreign invading antigens that have been able to get through our IgA defenses. They especially play a part in the destruction of large antigens like parasitic worms that are not easily phagocytized. And that's what that IgE molecule looks like. And that's all I'm going to do in this video. So the next video for this section of Unit 1, we're going to talk about antigen and antibody reactions and how we grade those agglutination reactions.